Assalamualaikum and hi everyone. So today, my group will be presenting about topic the bacteria. So what is bacteria? Do they good or harm to us? To know the answer, stay tuned till the end. So briefly, bacteria are the most common types of prokaryotes. They are simple unicellular organisms that lack of nucleus and also membrane-bound organelles. And the DNA of bacteria can be found in the region of the cells called nucleus. So bacteria can be found in every environment on Earth. In today's presentation, my friend and I will be presenting about the characteristic of bacteria, bacterial metabolism, and also symbiotic relationship. So without further ado, let's get started.
So now we're going to talk about the characteristic of bacteria cell. So there are two types of bacteria has been distinguished based on the structure of their cell wall. The first one is the gram-positive bacteria and the second one is the gram-negative bacteria. So bacteria is one of the domain under prokaryotes. Bacteria can be characterized based on their cell wall type, shapes, and their growth arrangement. So most bacteria cells are protected by a cell wall composed of the unique molecule peptidoglycan, a complex polysaccharide linked with amino acid. So bacteria have cell wall made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variation in osmotic pressure due to the different solutions they encounter, and it is their cell walls which prevent them from shrinking or swelling. So gram-positive bacteria has a very thick peptidoglycan cell wall, and of the 12 gram-positive bacteria, 6 are cocci and 6 are bacilli. So for gram-negative bacteria, it is surrounded by a second plasma membrane outside cell wall, often block antibiotic drugs, making infection difficult to treat. So gram-negative bacteria are more resistant to antibodies because of their impenetrable cell wall. So the differences between gram-positive bacteria and gram-negative bacteria is that gram-positive bacteria has a simple cell wall compared to gram-negative bacteria. Gram-positive bacteria has a thick peptidoglycan cell wall layer, while gram-negative bacteria has a thin peptidoglycan cell wall layer. Gram-positive bacteria has no outer lipopolysaccharide wall layer, while gram-negative bacteria has outer lipopolysaccharide wall layer. Gram-positive bacteria appears blue or purple in gram stain, and gram-negative bacteria appears red or pink in gram staining. So we could see the differences between gram-positive bacteria and gram-negative bacteria in this picture. Like I have mentioned before, gram-negative bacteria has a lipopolysaccharide and outer membrane. And you could see the thickness of the peptidoglycan in gram-positive bacteria is a little bit more thicker. Now we're going to talk about gram staining. Gram staining is a common technique used to differentiate two large groups of bacteria based on their different cell wall constituents. The gram stain procedure distinguishes between gram-positive and gram-negative group by coloring these cells red or dark purple. So there are 4 steps of gram staining. So in step 1 is where the crystal violet is applied, both cell walls affix the dye. Step 2 is where iodine is applied. So crystal violet iodine complex form inside cells, all one color. So in step 3 is where alcohol is applied. So a decolorizer such as ethyl alcohol or acetone is added to the sample, which dehydrates the peptidoglycan layer, shrinking and tightening it. Gram-positive remain the same. However, the outer membrane of gram-negative bacteria is degraded, and the thinner peptidoglycan layer of gram-negative cell is unable to retain the crystal violet iodine complex, and the color is lost. So we could see that in the step 3, where gram-negative bacteria become colorless. And in step 4 is where safranin is applied. A counter stain, such as a weakly water-soluble safranin, is added to the sample, staining it red. Since the safranin is lighter than the crystal violet, it does not disrupt this purple coloration in gram-positive cell. However, the decolorized gram-negative cell are stained red. So as conclusion for this gram staining is that the gram stain detects peptidoglycan, and since gram-positive bacteria have a thick multi-layered peptidoglycan, they retain the crystal violet dye. However, gram-negative bacteria do not retain the dye for 2 reasons. They have an outer membrane getting in the way of the crystal violet, and they lack peptidoglycan to retain the stain. So now we're going to proceed with the shapes of bacteria. So there are 3 basic shapes of bacteria. The wide variety of shapes is determined by the bacterial cell wall and cytoskeleton, and it's important because it can influence the ability of bacteria to acquire nutrients attached to surfaces, swim through liquids, and escape predators. So there are 3 common shapes of bacteria. The first one is Cocci, which is the round or spherical shaped cell. The second one is Bacilli, which is the rod-shaped bacteria. And the third one is Spirilla, which is the spiral shape or helical shape. So many bacteria grows in colonies. We say Diplo if it is found in pairs. We say Strepto if it is found in chain. And we say Staphylo if it is like grape-like cluster. So when you see these prefixes, it gives meaning to the type of bacteria.
Hi, today I will present about bacteria metabolic. First, we have three types of metabolism process, which is aerobic, anaerobe, and facultative anaerobe. For aerobic bacteria, the bacteria need oxygen to grow, which is the bacteria need oxygen to metabolize and produce the energy. The example is Pseudomonas and also Mycobacterium. And then we have anaerobes, which precisely is obligate anaerobe bacteria. This type of bacteria are unable to grow in the presence of free oxygens, means the bacteria metabolize without oxygen and also oxygen is poisonous for them. Some obligate anaerobe bacteria can survive up to 8% oxygen concentration in the atmosphere, and some of them only can survive up to 0.5% of oxygen concentration in the atmosphere. The example is Clostridium and also Bacteroides. And then we have facultative anaerobe. For this type of bacteria, they can survive both condition at the presence or absence of oxygen, because this bacteria can do both process, aerobic and anaerobe, depends on the presence or absence of the oxygen. And the example is E. coli and Staphylococcus. And then we have some disease that caused by this type of bacteria. First, we have aerobic bacteria, which is Mycobacterium bacteria. Mycobacterium is a bacteria that infect mammals, such as animal and human. And two types of Mycobacterium that infect human is Mycobacterium tuberculosis that cause tuberculosis, or we call it TB. And then Mycobacterium leprae that causes leprosy, or we call it hand disease, which is skin infection. And then we have obligate anaerobe bacteria, which is Clostridium. For Clostridium perfringens, it causes gas gangrene, which is a rare condition when the soft tissue doesn't get blood flow and also oxygen supply. And then Clostridium perfringens will infect the large soft tissue area and produce toxin and also gas. This toxin and gas will kill the tissue. And then we have botulism that causes by Clostridium botulinum. Botulism is a condition when humans eat a food that contaminated with botulinum toxin that produced by Clostridium botulinum bacteria, and will cause difficulty breathing, muscle paralysis, and even death. And then we have Staphylococcus aureus, which is facultative anaerobe bacteria. This bacteria will cause skin infection to human. And then we have autotrophic bacteria. Autotrophic bacteria, first we have we can divide by two, which is photoautotrophic bacteria and also chemoautotrophic bacteria. First we have photoautotrophic bacteria, which is the bacteria obtain energy from the sun. We can divide it by two, which is anoxygenic photosynthesis, which is this type of bacteria does not produce oxygen and just use photosystem 1 only, and have unique chlorophyll called bacteriochlorophyll. The example of bacteria is green sulfur bacteria or chlorobium, and purple sulfur bacteria Thiospirillum. And then we have oxygenic photosynthesis, which is the bacteria produce oxygen and use photosystem 1 and 2. The example is cyanobacteria, which is the type of bacteria have a characteristic of algae. And then for the second type of autotrophic bacteria, which is chemoautotroph bacteria, the bacteria carry out chemosynthesis by oxidizing inorganic compound to obtain energy by producing organic compound. The family example for us is Nitrobacter and also Nitrosomonas that contribute in a nitrogen cycle. Nitrobacter will oxidizing nitrate into nitrate in soil, and Nitrosomonas oxidize ammonia into nitrate. And last, we have heterotrophic bacteria, which is chemoheterotroph bacteria. This type of bacteria obtain energy by consuming organic substance from other source. First, we have saprobes, which is the bacteria feed off the remains of dead plant and animal and work as decomposer that break down organic material from that organism for the nutrient cycle. The example is E. coli and Spirocheta. This type of bacteria have characteristic like fungi. And then we have parasite, which is the bacteria live on or in other living organism and consume nutrient from the host, just like all parasites characteristic. And the example is Rickettsia and Chlamydia. And that's all from me.
Today I will be presenting about symbiotic relationships. First, biological interactions result when organisms in an ecosystem interact with each other. Today, we will be discussing about a long-term interaction which is called symbiosis. Bacteria and archaea form a symbiotic relationships where two different species live together in an intimate way. So here we have three types of symbiotic relationships. The first one is mutualism, and then we have commensalism, and lastly we have parasitism. The first type of symbiotic relationship is mutualism. In mutualism, both species will get benefit from the association. The first example, mutualistic bacteria living in human intestines release vitamins K and B12 that helps in produce black components. Mutualistic bacteria also helps to break down some substance in food that cannot be digested, such as fiber. And in return, the bacteria benefit by getting food. Second one is stomach of cows and goats. Mutualistic prokaryotes digest cellulose in plant, enabling this animal to feed on grass. In this relationship, both parties will benefit because the cow provides the bacteria a warm and moist environment with a constant supply of food, while the bacteria enable the cow to gain energy from the products of bacterial metabolism. Lastly, mutualistic bacteria live in the root nodules of soybean, clover, and alfalfa plants, where they produce atmospheric nitrogen to ammonia through a process called nitrogen fixation. For example, in nodule of legume, Rhizobium species convert atmospheric nitrogen to an organic nitrogen the plant can use, and in return, the plants provide food and shelter to the bacteria. Next, moving on to commensalism. It occurs when only one species benefits, whereas the other species is unaffected. For an example, obligate anaerobes can live in our intestines only because the bacterium Escherichia coli uses up the available oxygen. This is because obligate anaerobes can only survive in the absence of oxygen. This is the example of obligate anaerobes. Its name is Bacteroides fragilis. The last part is parasitism. Parasitism is a relationship between two species where one species benefits while harming the other's organism. For example, parasitic bacteria can cause disease, therefore are called pathogens. When gram-negative bacteria are killed by an antibiotic, their outer plasma membrane releases lipopolysaccharide. And here I attach the picture of lipopolysaccharide, which acts as a super antigen to overstimulate the immune response. So it may result to a high fever and a severe drop in blood pressure. Here are the examples of parasitic bacteria. The first one, Clostridium tetani bacteria. It can cause tetanus. And the second one, Shigella dysentery. It can cause diarrhea. During unfavorable conditions, bacteria can survive by forming endospores because endospores survive in the harshest of environments, desert heat and dehydration, boiling temperatures, polar ice, and extreme ultraviolet radiation. They also survive for a very long periods. Endospores formation allow the survival and dispersal of bacteria to a new place. Below here is the structure of endospore. And on the right side, I attach the endospore formation. First, DNA will duplicate. Then, a septum forms dividing the cell asymmetrically. Then, the large compartment will engulf the smaller compartment that will make the endospore start to form.
So now we're going to proceed with the bacterial diseases in human. So for the category of sexually transmitted diseases, for example, is gonorrhea. For respiratory diseases, for example, is strep throat or scarlet fever. For skin diseases category, for example, is impetigo acne, infection of surgical or accidental wounds and burns. For digestive tract diseases, for example, is food poisoning. For nervous system diseases, for example, is spinal meningitis. For systemic diseases, for example, is plaque and other diseases such as Lyme disease. Now we're going to proceed with cyanobacteria. So cyanobacteria are gram-negative bacteria with a number of unusual traits. They photosynthesize as the same manner as plants. They were called blue-green algae and were classified with eukaryotic algae, but now are classified as prokaryotes. They have other pigments that mask the color of chlorophyll and not only appear in blue-green, they can also appear in red. So you can find cyanobacteria in all kinds of body of water, but it's more common for them to grow in shallow, slow-moving or still water. This includes pond, wetland, and shorelines of freshwater lakes, streams, and rivers. The comparison between bacteria and cyanobacteria. So bacteria is comparatively smaller than cyanobacteria, and bacteria can be found in every possible places in Earth. However, cyanobacteria can only found in presence of sunlight and moisture. So for flagella, bacteria may bear flagella, while cyanobacteria flagella are always absent. For the composition of cell wall, for bacteria is glycolipids and peptidoglycan. For cyanobacteria is cellulose and pectin. For the reserve food for bacteria is glycogen, and the reserve food for cyanobacteria is cyanophysian starch. For the spore formation for bacteria is endogenous, while for the spore formation for cyanobacteria is non-endogenous. Bacteria take place by conjugation, transformation, and transduction. However, for cyanobacteria, the sexual reproduction is totally absent. So cyanobacteria usually reproduce by vegetative methods like fragmentation when the condition is favorable and water is available. So what happens if care is not taken in disposing of industrial, agricultural, and human waste? Cyanobacteria are ecologically important. When the amount of cyanobacteria in the water body increase, a dense mass called a bloom will form. Cyanobacteria blooms are natural. However, some human activities such as agriculture or a poorly functioning septic system can make blooms more likely. If care is not taken on those mentioned aspects, phosphate drain into lakes and ponds resulting in a bloom of this organism. The surface of the water becomes turbid and light cannot penetrate to lower level. When the cyanobacteria die off, the composer feeding on them use up the available oxygen, causing fish to die from lack of oxygen.
Okay, now we have come to the end of our presentation. Now we already know that not all bacteria are harmful. There are some bacteria that give benefits too. So we hope that everyone here gets new knowledge from our presentation. Bye bye!